So Schoology's open, yes. You see that all this, the summary of everything genetics is on there. Everything we've covered up to this point. The last thing that we need to cover is this, transcription translation, which could be included in that on that AP practice exam that you're going to get on Tuesday. We said yesterday that the exams can be all multiple choice. We said we're going to try to stick with the AP timing. It's going to be on AP Classroom. Am I saying that correctly? Is that what we all agreed to? Yeah. Okay. We said it was going to be a reasonable number of questions, but all multiple choice. And we all agreed that it was going to be something. We said like 50 minutes, but let's say we have 40 minutes, so it's 40 questions. 40 questions in a period on AP Classroom, so you're going to need your computer, need your charger and all that. Everybody good with all that? Okay. So let's go ahead and start talking about then transcription and translation. Did anybody remember what transcription and translation are? I can't see you if you have your hand raised, but I'm assuming not, since usually you guys start talking. Two processes that are required to be done when you're trying to express genes, when you're trying to make a protein. So you have a gene, a DNA molecule. You have a section of the gene here, from here to here. This part would be a gene. Genes can be very long or they can be very short. By the way, I did post 2 10-minute videos in your assignment for this transcription translation. Each of them are going to explain it a different way. One is Khan Academy. The other one is not a Khan Academy. And then, of course, we're doing it here ourselves as well in class. So a gene can be defined as a sequence of DNA, right? DNA that is expressed. In other words, that becomes RNA, and then that could, and mostly does, become a protein. All right, so that's what a gene is. So those alleles that we've been talking about in genetics, and big A, little a, dominant, recessive, what we're talking about is a sequence on your DNA, in your nucleus, because that's where DNA is, in the nucleus, in a eukaryote. Again, a eukaryote. It's in the nucleus, is where the DNA is. And that gene is that part of the DNA that gets turned into RNA, and then it gets turned into a protein. So turning into RNA, we call that transcription. So transcription is when we take and we make transcription is when we take DNA and turn it into RNA. The reason is that DNA is in the nucleus of the cell, and the ribosomes are here in the cytoplasm. They're in this part of the cell here. And so the DNA cannot get out of the nucleus. So the message to make the protein is in the nucleus. But we have to find a way to get that message out. And we use that, the message coming out is known as mRNA. So RNA acts as a messenger in this case. RNA has several roles, but mRNA is the messenger. We have to take the message from DNA and turn it into RNA. What message am I talking about? A sequence of letters. A, T, G, C, A, T, T, etc. That is DNA, as I've taught you before. T is found in DNA, so thymine is T is only found in DNA, and it needs to get translated or converted. And we're going to use base pairing. We use base pairing. Life on planet Earth uses base pairing. So A should match with what base pair? T, correct. But this is not going to be matched with DNA. What's it going to be matched with? RNA. It would not be a T. It would be a what? U. So we'd have U. Then T matches with. You just said A matches with what? T. So what does T match with? A matches with T. That's it. So T matches with A. That's it. I understand where you might be confused, so get it straight, okay? G matches with what then? C. And C matches with G. That's in DNA. But in RNA, A matches not with T, but with U, and G still matches with C. Are we clear? Right. So that's the base pairing. That's what we call base pairing. Glad we could clear that up. So it's this ATGCATT is going to be transcribed. Right? It's going to be the message will be rewritten into another code. So A goes to U. What is T? What is T going to be matched with? A. Because there is A in RNA. G gets matched with, and C with, and then we have U again and A. So this is what is going to be on the RNA. So this is the message or gene, the beginning of the gene in DNA, and this is the message or beginning of the message in RNA. Does that make sense? Is there any confusion? Do I need to go back and explain any words that seem like it's ambiguous or you don't remember? Okay. By the way, again, in an attempt to try to reduce the amount of time you're spending at home reading, I've taken the liberty of posting in this assignment a summary of all this in written form off adapted from a web page. I basically copied and pasted the explanation of transcription translation with pictures. It should be no more than 5 or 10 minutes of reading, 2 or 3 3-minute readings. That should reduce it from having to read the whole chapter. I'd rather you read the chapter, but y'all tell me you need less stress at home. You have you don't have time to spend an hour reading at home, so this should break it down a little easier for you. Are we good with this idea of genes and this idea of transcription? What's this word that we're talking about here? You must know the difference. You have to know the difference. This is transcription. Is everybody good with that? All right. So then this message, after you've transcribed it, by the way, we should talk about an enzyme that's involved in this. It's a complex known as RNA polymer ACE. And we talked about it before last semester. It makes a polymer. ACE means it's an enzyme. ACE means it's an enzyme. And polymer, obviously, at least I hope it is obvious now, that it's taking a bunch of monomers and putting them together into a polymer. And of course, RNA is ribonucleic acid. So this is an enzyme that makes RNA. And you need to have some other supporting enzymes that help unwind the DNA. You need nucleotides, RNA nucleotides. In case you forgot, RNA is a ribose sugar, ribose, not deoxy. A nucleotide has a phosphate on it and has a nitrogenous base. The nitrogenous base is either A, T, G, or C if it's DNA, but in RNA, it's either going to be A, U, G, or C. That's what goes here. A, U, G, or C in RNA. That's a nucleotide. So you're going to put a bunch of these together. It's actually going to be a triphosphate, a whatever nucleotide triphosphate, so that you have the energy to put them together. The polymerase does this, puts them all in order. It reads the DNA. The double helix opens up. And the RNA polymerase lands, starts to read, and then starts to make RNA. It reads the DNA, reads the message on the DNA. A, where it sees an A, it puts a U. I say, yeah, when it sees a T, it puts an A. It sees a G, it puts a C, etc. And it goes down the line reading. All right, that's basically the process, and we'll look at some details in a minute. So that's an enzyme, that's a gene, that's transcription. Now, once you get an RNA, you get an RNA molecule that's pretty much the length of the gene from beginning to end. You have to do some what we call post-transcriptional modifications. So let's break down this term. What does post mean? After, right? So post means after. Transcriptional, what do you think that means? After it's been transcribed. So after you have the RNA, right? So you're starting with RNA. And what do you think modifications mean? Changes. So after transcription, we're going to make some changes. You're going to have the RNA, and then you're going to have a bunch of A's added to the end. A poly A tail. We call it a poly A tail. You're going to get rid of. You're going to have these things called exons. You can get rid of the introns, first of all, which on the surface, it doesn't make sense. Introns are getting cut out. So there's going to be parts of this that I'm going to label I, and I'm going to, in fact, put it in different color as well. These parts here of the DNA, RNA, they're going to be removed. They're going to be removed. And so now the RNA is shorter. It's going to put a 3' cap on it, and it's going to add a poly A tail. So it's shorter. The message has been changed. And here you keep the exons, I should say. So the I is the introns. The exons are in green. You're keeping the exons, and you're putting them together. So now you have a shorter message, and it's called. This is what's called mRNA. Now we have mRNAs, the messenger. But you had to do 1 and then 2. You had to take out the introns. Then you had to add the tail and the cap for it to become an mRNA. All this is happening inside the nucleus. in a eukaryote. How do you feel about that? Is that okay? Is that too much?
Could you go back some about the introns?
Sure. I don't want to get into details, but there are messages, there are chemical signals. Let me start from the very beginning. You have a gene. It's on the DNA. There it is. And it's this enzyme called RNA polymerase is going to start reading it. And wherever it sees an A, it's going to put a U. Wherever it sees a T, it's going to put an A. Okay? It's the base pairing. Got it? All right. Then it's going to fall off at the end, and you're going to have this copy of this message, copy of the gene. All right, so it's a copy of the gene in RNA. Everybody agree with that? It's the message in RNA. Okay. This is all happening in the nucleus. The enzyme that's doing this is called RNA polymerase. I put it up there. There it is. But we're not done. The message has to be changed. The changes are called post-transcriptional modifications. Several changes have to happen. Introns have to be cut out. Now, that doesn't seem like it makes sense on the surface. Introns cut out, but exons are being left in. How does that make sense, right? But exons mean expressed. So they're the part of the message that are expressed. So I don't like the term because it's always confusing to people, but that's the term that's used. So we're keeping the exons and we're removing the introns. So by definition, the message is shorter, correct? And we've changed it. Then we add this poly A tail. Again, we don't have time to get into why we're adding poly A tail. We add a poly A tail and you put a 3' cap on it. Are we good with that? And that's called the mRNA. That's it. That's what you got to know. That's the part of the message that's going to leave the nucleus. Bless you. You got that? How do you feel about that? We got it? Yeah? All right. So that's the part of and there's no confusion. All right. So now again, we have the nucleus has a nuclear envelope. It's attached to this thing called endoplasmic reticulum. I'm going to put this endoplasmic, I draw it as a line, but it's a giant membrane system. It's a very large membranous system. It actually surrounds a lot of the nucleus, has a lot of ribosomes on it. These are ribosomes, studded with ribosomes. That's why we call it the RER, rough endoplasmic reticulum. And then it turns into the smooth ER, SER, without the ribosomes. Ribosomes are involved in what? What do ribosomes do? They make the proteins. So a ribosome, everyone should know what that is actually made of RNA. It's not a protein. It is made of RNA. So you have two parts. You have a large bit and a small bit. Small. We actually call them, amazingly difficult words, large and small subunits. So this ribosome is made of two parts. The messenger RNA is going to go in between here, between the large and small parts. And this is going to be reading it, and we'll talk about reading it in just a second. We'll use the actual document to look at reading. It'll read the message 3 nucleotides at a time. The nucleotides are called codons. 3 nucleotides at a time. And what could be in this nucleotide? Could be A, could be G, could be C, whatever. Whatever's in there, it's going to read it 3 at a time. So every word, for the ribosome, every word has 3 letters. Is that clear? I I hear silence. Although I do enjoy the silence, I worry you're not paying attention. But all right, I heard yes. So it's called a codon. Codon's a triplet code. It's done in threes. There are also ribosomes throughout the cell, throughout the cytoplasm. Free-floating ribosomes that are used to make proteins that are in the cytoplasm. Does that make sense? So if the protein is if the protein's here in the cytoplasm, then you're going to use then the messenger RNA is going to come out here. It's going to be used here. If one of the post-translational modifications, if part of the code can be a series, a little message that tells the cell it stays in the RER. The messenger RNA will bind to these ribosomes and will get sucked into the RER, rough endoplasmic reticulum. In that case, when they has that message, the protein is going to go from the RER and to the SER, from the SER to the Golgi body. That's what this is, Golgi body. And from the Golgi, it's going to end up going to the cell membrane. It'll fuse, and the protein will go out of the cell. Or it'll fuse with some other organelle like a lysosome or a peroxisome. And so the protein could end up here in a vesicle or in some organelle, or it could be put out of the cell, right? What are examples of proteins that are put out of cells? We should know a bunch of them. Who could tell me a couple? What are some proteins that are put out of the cell? Nothing, huh? No proteins that you can think of that get put out of a cell. Just not feeling it today, huh? Nothing? You can't think of anything the cell will put out? Why don't you guys look it up? Take a minute, look it up. So I can't be the only one talking. I'm gonna end up exhausted, and you're gonna end up not learning as much. Why don't you tell me? Why don't you use that phone instead of texting or talking or TikToking or whatever? Could you look up? She's not even listening to me.
Who, me?
I was talking to her, but you too, yeah. Look up.
Google.
Oh, you're Googling now? Oh, okay, my bad. I saw you texting earlier. I thought you were still texting. What kinds of proteins are put out of cells? What kind of proteins go through exocytosis? Sounds like we have to have a quick review. So proteins that go through examples that I've mentioned in class before, they're put out of the cell. And I'm sure you know some of these. Insulin. You've heard of it before? I know because I've said it in class. Glucagon. Both of them help regulate sugar. What are some other things that leave the cell? I know you learned this. I taught it to you. Mucus. Snot. Insulin, glucagon go into the blood. Mucus, obviously, mucus cells that line your respiratory system and digestive system will, given a certain stimulus, will bind to the membrane, the cell membrane, the lining of the respiratory tract or the intestine, and release mucus to the outside. That's how mucus works. Digestive enzymes. Amylase in your saliva. Saliva itself has mucus in it and proteins in it that your cells release. Amylase is one of those enzymes to help digest sugars, help break down sugars. You have enzymes throughout your digestive system that release enzymes from inside the cell into your digestive system. Those are just a few of the examples of cells that are making proteins to leave the cell. Other proteins have to stay inside the cell. So proteins like in the cytoskeleton or that make up other structural components of the cell have to stay inside. So those are made here. This system, you will see it mentioned on AP in the textbooks, in the readings, and on the AP exam, you'll see something called transmembrane system. And what they mean by that is that the mRNA and proteins that are made within this system from one membranous organelle, the rough ER to the smooth ER, to vesicles, to the Golgi body, to more vesicles, to fusing and leaving the cell. By the way, we've done this already back in October. When we see this pathway of proteins leaving, we call it the transmembrane system for exocytosis. This is how the path it follows. Also moving this way into lysosomes, into peroxisomes. That's also part of the transmembrane system. But proteins that are made to stay in the cytoplasm, enzymes that are used in the cytoplasm to help build cytoskeleton, for instance, they stay here. They're made by free-floating ribosomes. Okay. But that's the basic idea. Has everybody got that so far? Is that okay? Were those good examples of proteins that are released outside the cell? Are they familiar to you? Yes, I see some nodding. Okay. So let's go ahead and take a look then at our assignment that we have today. Oh, you zoom back out for something? Okay. Are we good? All right. All right, so if we look at our diagram, and you could read, I don't think we need to read, but why DNA? Why do we have to do why do we have to change the message to RNA first? And I already said it. It says it right here as well. You can read it in a paragraph. But DNA is here. We have to transcribe it into mRNA because it has to leave the cell, the nucleus. It has to leave the nucleus because the ribosomes are out here. Does that make sense? You can't make a protein if the message is in DNA, if it's in the nucleus. You have to unwind the DNA. RNA polymerase has to land, has to make the RNA. You have to change the RNA. The RNA has to be made shorter. You have to have poly A tail, put a cap on it. Then it has to leave the nucleus. Are we good? All right. There's another kind of RNA you can see here that's called transfer RNA. And all it does is it brings, and it says it right here, brings the amino acid to the ribosome. That's the whole job. It's made of RNA. By the way, I need to mention this because this is a confusing point for a lot of people. Not all genes make proteins. There is a gene for tRNA. The only thing it makes is it only makes RNA. All genes make RNA, but not all genes make proteins. Some genes just make RNA, like transfer RNA, like ribosomal RNA. There are genes that make those. Is that clear? Yes or no? I see like 3 people on their phones. I see a couple people saying yes. Stanley, hey, how's it going? Nice speech, by the way. Sorry, I was only playing around when I booed. All right, so tRNA brings amino acid. It is okay. So we understand that not all genes make proteins. So each amino acid. Now this is a key idea as well. Before we go any further, each amino acid, and I could list them. We could say things like alanine, tyrosine, proline. There's a lot of them.
These look like stuff that be on like bottles.
That's exactly, they are on bottles.
Yeah.
They are, yeah. These are amino acids that you'll find listed, in, especially if you do any protein shakes, any kind of GNC training, any kind of health drinks or whatever. You'll see high in tyrosine, high in alanine, high in this, high in that. There are how many amino acids? There are a lot of amino acids. How many? Look it up. How many amino acids are found in the human body? Did you find 21?
20
20 amino acids. 20. I always want to say 21 myself, by the way, so I don't blame you for saying it. But there's 20 amino acids that are, and often I do, by the way. If you ever hear me say 21, it's a mistake. So these are what are being carried. They're being carried by an RNA that, oddly enough, looks like a T, to me, anyways.
There's a diet person. What do they do?
They make proteins.
They make protein.
They are the monomer. You're not even in this class, but they are the monomers. They are the monomers that you put together to make that thing we call protein. So your hair, your nails, your skin, your muscles are all made of this. A bunch of these put together in long chains. All right. I better plug this in before it's too late. I wonder if I can use this. Oh, I can. That's nice. All right. So tyrosine is just one example. The question is, you have this mRNA, and you have a sequence in 3s. We said there was in 3s, right? Yes? Yes or no? Yes. Okay. You got a big ribosome here that, by the way, has these three little spots where the tRNAs fit, by the way. The tRNAs are going to fit in here. We'll look at it in a minute. You have these 3 spots. You have this message AUG. Well, how does a ribosome read that message and make sure that the right tRNA gets put in there? There has to be a match, right? That's how this reading happens. Is that correct? You have to match these. So this RNA is going to read AUG, and it's going to allow the tRNA that has the right match. What would be the matching letters for AUG? It'd be U.
Oh,
UAC. So UAC is called the anticodon, and it's attached to tRNA that has an amino acid called methionine on it. All right, so it's a bad drawing, but that's what's going to be because I don't want to redraw this. So UAC is at the end. This is all RNA, right? This tRNA is made of RNA. Right out here, it's going to have methionine, and on this end, it's going to have U, it's going to have an A, and then it's a C. Now, this part, so it's going to come in, it's going to fit into one of these slots that the ribosomes have, the UAC will match up with AUG, and that's how the ribosome knows that that's the amino acid that goes in that position. And it's going to continue to read. It's going to continue to read in threes. AUC, maybe the next is UGA. This part down here is known as the codon. The message on the mRNA is known as a codon. They're in threes. The message that's in blue up here, that's on the tRNA, is known as the anticodon. The anticodon matches the codon. It literally, like a secret, like a spy matching the letters, it's going to match AUG to UAC. It's going to match ACU to UGA, etc. Well, this only works if you have a specific set of amino acids that are on a specific tRNA. So this is obviously a freshman biology honors activity. I thought it'd be a good intro rather than jumping right into the AP Bio one. Is that okay with you? Since today's Thursday, then Monday we could do a more difficult one. Is that all right?
Yeah.
I know some of you will be missing on Monday. You'll have to, because there's a field trip.
We don't actually have a case in the same district. It ends this Friday.
Oh, okay.
And that's fine.
That's fine. So everyone will be here on Monday, and we can go over this in a more detail, in a more difficult case study on Monday. But you do that. Okay. So again, to clarify, G goes to C in DNA, T to A to T, G to C, C to G, T to A to T, A to T, C to G, C to G, T to A, T to A. That works because that's DNA. DNA and DNA, they match up. Double-stranded, long. Now we're making a copy of the RNA. We're reading T goes to A goes to U, not T. C goes to G. Then C goes to G again. A goes to U. T goes to A. C to G. G to C. A to U. T to A. T to A. G to C. G to C. A to U. A to U. Matching, matching, matching. Complete the statement below. In DNA, adenine, also known as A, always pairs with what? T is correct. Thymine is correct. Cytosine always pairs with what? G, which is guanine. Guanine. Don't worry about it. Just G. Just know G. You don't need to know the words. A with T, G, C, and G. That's it. Different nucleotides. Again, back from first semester. Fill in the missing bases of double-stranded DNA in the three examples. Already did that. There they are. In mRNA, A is always paired with what? U or uracil. And C is always paired with G or guanine. Fill in the missing bases. We just did that. Okay. So going from DNA to RNA is known as transcription and happens in the nucleus. Now we're going to make translation. Now we're going to do translation. And there's details here, as you can see here. Second step to making a protein. So here to make a protein, we're going to use this chart, this codon chart. there's two charts because I don't know which chart they're going to give you on the test. I know we have 2 minutes left. I'm going to use a minute of it. I'm not sure which of these two charts you're going to need to use, but you need to know how to use both of them. You'll have both on my test that you're going to get on Thursday. So in this chart, you start with the center one. If you're not looking at the board, you're not understanding. So you start with the center is the first a letter of the codon. Remember, codon is 3 letters. So let's say A-U-G is the first 3 letters. That's the first codon. You start with the first one, A, then you go to U, and then you go to G. A-U-G is methionine. Do you see that? This is the amino acid on the outside of the circle. The first position is in the center of the circle. The second position is one of these in the second circle. In the third circle is the third position. And then you finally have the amino acid. AUG is always methionine. AUU is always isoleucine, etc. In this chart, you do the same thing, except they label it first. This is the second letter, and this is your third letter. So A, U, and G, AUG, A, U, and G would give you methionine, also known as start. So that's how you read both of those charts. I'd like you to use a pencil and try to solve the rest of this at home, and we will finish going over it tomorrow. All right, I'm moving on. And so you have AUG was methionine. What the next one's GUA was what?
GUA was
VAL, valine, valine. GCU is what? Alanine, ALA. AAC is what?
ASP.
What is it? ASP is fine. CUU is what? LEU. Leucine, it's fine. What about AUU? ISO. And GGC? GLY. CUA? LEU. UUG? PHE. And GUA? VAL. All right, that's how you read it. That's how you translate it. That's it. And when you're doing this, you have to translate it exactly what we just did. However, just so you understand, there is the large subunit has 3 spots. Large subunit on a ribosome has 3 places. That's horrible drawing, but that'll be fine. One, the mRNA gets read here. So here you're going to have AUG. Each one, maybe this is CCU. AUG, this is going to initiate here. This is where actually the bond is going to make, and this is where it's going to get expelled. And the tRNA is going to come in. It's going to have some kind of amino acid, methionine, let's say, attached to it. On the opposite, it's going to have, on the opposite end, this is an RNA molecule. It's going to be the opposite of AUG, which will be UAC. And it's going to match AUG. UAC is going to match. Again, forgive the horrible drawing. And it's going to have this methionine here. Methionine is going to get linked to the next one. What's CCU? What is CCU? So it's GGA. And what's CCU? What amino acid has CCU on it? Or rather, what codon is associated with CCU? What amino acid is associated with CCU? What is it? Proline. So now when these two get ratcheted, they move over. So they're going to move in this direction. The mRNA is going to move in that direction. As it moves over, what's going to happen is the mRNA is going to move over. It's going to move in that direction. And then methionine is going to have a peptide bond to proline. And then the next amino acid in the chain gets put on. And the next one, they just keep getting made here at this. They get expelled here, the bonds form here, and they just keep making a chain. It happens very quickly. This is the larger subunit, then there's a smaller subunit underneath. That's a sketch of the process. I can't imagine they're going to ask you a question about initiation, termination. This would be the termination set where it gets kicked out, but I can't believe that they're going to ask you those questions. Mr. Mendoza's room, may I help you? Okay, thank you. All right. Are we all good with that idea? So the translation is pretty straightforward. Find a ribosome, start an MET, read the chart, put them in. No problem. You go ahead and do this and answer the questions. Please note, this is mRNA, this is the amino acid, and these are the traits. Where am I going to find the traits? The traits are here on this chart. These are all the traits, all the possible traits. How do you know what traits they have? If you have these three amino acids together, you get hairless. If you have these three amino acids together, you get hairy, etc. It's just like sickle cell. You have a certain set of amino acids put together, you have sickle cell hemoglobin. You have another set of amino acids together, it's normal hemoglobin. But here you're just looking at some really basic traits. I'm going to pause here. I'm going to ask some people to come up here.